Hello and welcome back to all of my friends and my students. Today we are here to discuss the tricks and mnemonics of Krebs Cycle of TCA Cycle. I am your biology mentor Bhaskar sir. You can read about my motto and all details in the about section of these channels. So I was thinking this particular topic TCA, the Krebs Cycle, for that Hans Adolf Krebs got Nobel Prize in 1953. It is so important that in each and every examination, every year in NEET and other All India Comparative Examination AICE exam, this question appears, Krebs cycle or TCA cycle. Sometimes in examination in 5 marks you can have this question. You have to write down the sequence of citrate, alpha ketoglutarate. Exact sequence has to be remembered. But the most important thing is the logic. How this cycle is created, in which step substrate level phosphorylation takes place, which step CO2 get released. So to understand this total method, we shall go for a galvanization technique. So galvanization technique is the combination of two things, mnemonic tricks and the logic. So this is how we can, because there are trillions of topic, so you cannot make trillions of mnemonics to remember those things, because you have to remember the mnemonics to produce the answer. So that is why we have to understand the logic, and combination of logic and mnemonics is known as a galvanization technique. So let's come to the understanding first. In 1937, Hans Adolf Krebs discovered this TCA cycle. It happened in the mitochondria. So he was studying the pigeon, he was studying a bird breast muscles, and he found the rate of this energy production along with the OX pathway. So this is very high. So he discovered that one in the pigeon breast muscle, 1937, and in 1953 he got Nobel Prize for this discovery of the cycle. Cycle means it goes continuous in this direction, and in one step you have the production of GTP and ATP, and some step you have the production of that one CO2, and total 10 enzymes are required. That is very important. Total 10 enzymes are required. In 4 step you can see the dehydrogenation reaction and 2 carboxylation reaction. So 4 dehydrogenations and 2 carboxylations. What should be the first step to remember it? First, you know that 10 enzymes are there. So first you draw 10 arrow. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. This is the first step. So this is the logic, 10 enzymes. That means 10 arrows are there present. Now you have to use this mnemonic, that CIO alpha SSFMO. This is called word mnemonic. CIO alpha SSFMO. Citrate, cis-aconited, isocitrate, oxalosuccinate, alpha ketoglutarate, succinyl-CoA, succinate, fumarate. You can call as a fumaric acid. So fumarate, malate, and oxaloacetate. So now this first 4 is 6 carbon compound. Correct? How it is coming, we will discuss it. And this one is a 5 carbon compound, and rest of the things from succinyl-CoA to the oxaloacetic acid or oxaloacetate is 5 minus 1, 4 carbon compound. What does it implies? It implies, as you are having 5 carbon here, so these two regions is the place where CO2 is being released. So this is the step where decarboxylation takes place. Most important enzyme, decarboxylases. So CO2 is ejected out. So you can answer in which, or name the steps where CO2 being released. That is in between oxalosuccinate. This is oxalosuccinate. This is your alpha ketoglutarate. So in between these substrate, in these two chemicals, one molecule of CO2 is released. So the number is being reduced to 6C25C, then 4C. So alpha ketoglutarate to the succinyl-CoA, and here you can see the one release of the carbon dioxide gas. So now you can remember the number of carbon and the region where CO2 is being released. First you note this concept down, then you can put the names here. So here is citrate. Now the structure of citrate, if I draw here, I think you can see The name of the cycle is known as a TCA cycle, that is tricarboxylic acid cycle. That means tricarboxylic groups are present. What is present? Tricarboxylic group is present. So draw three carboxylic group, then you connect it with the carbons. OH is present here, then CH2, and here also CH2. So when the cis econidate is formed, the one water molecule is being released from here. So now you can redraw it in this way. So this is your OH group. So when the one water molecule is removed, you can get a double bonds over here. So now this is the citrate will be converted into cis econitrate with the help of econitase enzyme. I will discuss the full chart with the name of the enzyme, because you need not to remember all the name of the enzyme but the concept. So logic is that one water molecule will be removed, so cis econitrate will be formed. Then you have a isocitrate. Then isocitrate will form oxalosuccinate. I am using the abbreviation because of the full name we will be having. So just we have to remember this tricks word mnemonics. You can remember it by making your own mnemonic. This is not standardized or universal. So that is why I appreciate to remember or understand the logic than mnemonics. So you can remember cute college Indian of Alpha School saved forest and mountains. Correct, in this way, or you can make your own mnemonics, but logic here, that is very important. So this is called galvanization technique. So alpha, before alpha all are 6C, alpha after the alpha all are 4 carbon. So in between these, release of CO2, hence the number of carbon being reduced here. Now oxalosuccinate will be converted into alpha ketoglutarate. Correct, alpha ketoglutarate. Whenever you say it, that means it is this. When you say malate, that means this group is present. When you say malic acid, this will be converted into COOH. So that will be acid. Correct? So alpha ketoglutarate, then succinyl-CoA. I will come from pyruvate to the acetyl-CoA. That's I am following the reverse technique. So now succinyl-CoA, then succinate. I will share you the structure with another PPT. Then fumarate, or you can say fumaric acid. So fumarate, this is the structure of the fumarate, like mirror image. CH, then CH, COO minus. Correct, you can see a bond over here. Then fumarate, then malate. So these are all 4 carbon compound, because after the alpha ketoglutarate, because there is a one release of CO2, and before before this and after this there is a release of CO2, definitely it will be then oxaloacetate. As in the C4 cycle you can see oxaloacetic acid, so it is the oxaloacetate. Now this is your 4 carbon. So how it get converted to the 6 carbon citrate? It is simply from the pyruvate that converted to acetyl-CoA, coenzyme A, that is 2 carbon compound. Now the combination of 2C plus 4C goes to A, will be having 2C. Oxaloacetic acid is a 4C, so it will be 6C. It is like that. You are trying to start a business, you need 6 crore rupees. So you are taking 4 crore from oxaloacetic acid, so you have to return it back, and you are also using 2 crore, that fundamentally you have only 2 crore rupees. So acetyl-CoA, so you have to depend on the 4C that has been taken from the OAA. Now you can make the 6C. In the meantime, you have done your job, and finally you have given the 4 carbon compound back to the system. That is why you have to maintain the cycle. Okay, now I have partially cleaned the board. Now you can add one more concept, in which step substrate level phosphorylation takes place. So one very important enzyme, that is synthetase and synthase. For example, here one synthase enzyme is required at the first step. Citrate ECR will be sharing, but here whenever you are trying to create ATP or GTP, so you need a synthetase. So the synthetase enzyme is associated with the conversion of this state, succinyl-CoA to the succinate. What is succinate? Succinate is this CH2COO minus CH2COO minus. So if you say succinic acid, it will be CH2COH, CH2COOH in that way. So succinate, and you have a succinyl-CoA, the CoA enzyme A, that group is removed. In between you have production of 1 GTP, and that 1 GTP is equivalent to 1 ATP. So in this particular step only we have to remember this step in between succinyl-CoA and succinate. What production of ATP is done, equivalent to the GTP. So this step is the called as substrate level phosphorylation. Phosphorylation means the production of ATP. So whenever you have a ATP, it being phosphorylated, that means one phosphate is added. So the word means substrate level phosphorylation, means when these two substrate being converted to from one to another, in between you have a production of 1 ATP. This is the entire pathway and the main goal of the total reaction. So that I think definitely it will help you to remember the logic and this galvanization method. Let's check out the original figure and name of the enzyme. Okay, I'll continue.
Okay, now we are here to share screen with you. So as we know, in the eukaryotic, the citric acid cycle takes place in the matrix of the mitochondria, and this cycle repeated in the aerobic respiration. It goes on and on. Just we need to revise and repeat the process. As we know, repetition is the raw material for the memory. So just we have to revise and repeat the things. So let me take one highlighter so that I can focus on it, on the molecules that has been given. So first, 2 carbon compound that is being produced from the reaction of the pyruvic acid and acetyl-CoA. So this is your CH3COS-CoA, acetyl-CoA, that reacting with the oxaloacetate that is already present in the matrix. So that is your 4 carbon compound. The enzyme that is required subsequently, that is citrate synthetase, that converts the oxaloacetate to the citrate, that is a 6 carbon compound. Citrate or citric acid means it should contain 3 COOH group here. So as it is a citrate, H has been removed, otherwise 3 COOH group is present. So hence the name is a tricarboxylic acid, because in the citrate or citric acid we are having 3 carboxylic acids. Then aconitis enzyme is important with the help of iron Fe+2 as a metal cofactor, and water molecule is being removed, and you can see the double bond formation. So as already discussed, cis aconitide is formed. You just focus the structure, how the things are happening. So this is your 6 carbon compound. The moment one CO2 molecule is being released, now you can see isocitrate is being formed. Now the moment the CO2 is being released, the number of carbon is reduced to 5. So the first 5 carbon compound is a alpha ketoglutarate, that is your 5 carbon compound due to the release of CO2 during the aerobic respiration or oxidative phosphorylation in the mitochondria. Next, you need ketoglutarate dehydrogenase, and you need the cofactor NADH-CoA. So many factors are required, and again one CO2 is released, so the number of carbon will be reduced to 4C, that is a succinyl-CoA. Again, one ATP molecule is formed here. So again we have to put some stress this step. So succinyl-CoA synthetase means ATP is produced. So from this transition, succinyl-CoA to succinate, substrate level phosphorylation takes place. They can ask you at which phase substrate level phosphorylation takes place. You can say during the conversion of succinyl-CoA to succinate, 1 GTP is produced, and 1 GTP is equivalent to the 1 ATP. So at this moment, substrate level phosphorylation is very important term that takes place. Otherwise, you can see in other phases, no where ATP is being produced. If it is 2 is there, NADH H plus can be produced, but this is the only step where you can see the substrate level phosphorylation. And again, FAD converted to FAD is to subsequently succinate converted into fumarate. So COO minus, CH, COO minus. This is a mirror image. If you take left and right or up and down, is same. So fumarase enzyme will convert it to the malate. COO minus, 8 means COO minus, and if you say malic acid, it will be COOH. in these two places. Again, this cycle continues to through oxalic acetate. Again, it will bind to the acetyl-CoA. This cycle continuously goes on and on. Name of these enzymes are not that much important for exam, but for understanding purpose, we very much focus on these things. And manganese and iron also the metals that are required during this process, and we focus on the step where ATP is produced and NADH H plus are being produced. With the help of subsequent repetition, you can remember the things in a better way. I think definitely these things will help you. Thank you for watching.